Very good. So thank you very much, everybody, for joining in today. And it's a great pleasure for me to be able to present this award lecture where I have kind of had a hard time deciding what I would present. But I thought I would try to give you a little bit of flavor for the way we conceive chemistry and the way we try to really embrace a multidisciplinary program in my group. There is always this common thread, which are transition metals. That's kind of my passion and my education. But I hope you will see how translational transition metal chemistry can be. So as it has been already mentioned in the introduction, part of our program is focused on the idea of utilizing organic molecules to interrogate biological systems. Part of our program deals with natural product synthesis, and we try to there not only synthesize molecules, but develop them into chemical probes so that, for example, in the case of iridium-meteorites, which are a target we have been involved with, we can try to unravel the cellular targets, which turn out to be actin in this particular case, and this open up a new field in the group dealing with trying to develop probes that would reversibly bind to actin and therefore could be used as cytostatic molecules. Of course, nature brings a lot of inspiration in the form of secondary metabolites, but we are also keenly interested in utilizing computational tools in order to educate our design for small molecules. And this is a work that my group does since many years with a computational biophysicist colleague here at the University of Zurich, Professor Amedeo Kaflish, and we are in the last few years very active in the field of epigenetics. Now, you see, this is the part that somehow connects organic chemistry and maybe a little bit of chemical biology, if you want. But a large fraction of my group is keenly interested in transition metal catalysis. And also we have a keen interest in mechanistic understanding of catalysis. So how interrogating reaction intermediates, we can gain some additional insights that might help us into developing new tools, new transformations not covered thus far. And in recent years, while gold has always been kind of our preferred metal to work with, we also ended up finding very interesting transformations that utilize radicals as kind of the key intermediates. And intersecting radicals with metals have become very recently in my group kind of a main theme as well. So in my lecture today, I will try to bring these two, in principle, completely unrelated topics and bring them together for you so that you can see how our, so to say, medicinal chemistry program can intersect with late transition metal catalysis. So in order to do that, I have to give you a very, very brief introduction on one of the fields that we are active on, and this is the field of epigenetics. So for those of you that are not so familiar with this word and what epigenetics is, let's suffice to say that epigenetics is the field that deals with mechanisms that regulate the expression of our genes. So traditionally, we understand that our phenotypes, our lives are determined by our genes, by our DNA, our genetic information, but there are huge mechanisms of control of those genes, when those genes are going to be expressed, to which extent they are expressed, when they have to be shut down, and all these mechanisms that control the expression of our genetic information is what we call epigenetics. And it's indeed epigenetics and all these complex mechanisms of control, the ones that explain the fact that even if we share 90% of our genetic information with animals like bees or other insects, we are phenotypically very different. So as you can see, genes are not the only determining factor in how we look like and how we function, but all these other mechanisms that are going to regulate gene expression. So this is, so to say, I mean, these are nice words, but at the end of the day, all these complex mechanisms of control can be boiled down to chemistry. Reactions that take place in the histone tails, so in the amino acid residues of the histones, which are the proteins that wrap up our DNA so that our DNA can be nicely packed into the cell nuclei. So histones, as I said, are proteins that contain a lot of positively charged amino acid residues, such as, for example, lysines. And there are mechanisms carried out by enzymes by which these positively charged residues can be acetylated, and therefore they become neutral. And by becoming neutral, then the nearby areas of the DNA become more exposed and therefore prone to be transcribed. The proteins that take care of this acetylation, sometimes also methylation pathways, are proteins that we call writers. It's clear that in nature, for every functional group or every process that you have to have the corresponding counterbalance. So there are other proteins that are in charge of erasing those functional groups and then restore the system to the initial stage. So the erasers will remove this acetyl group and will reveal again the positively charged amino acid residues. And then there is a third group of proteins that are going to be the ones recognizing that these residues have been acetylated and that therefore there has to be a cellular response, right? And these proteins are called readers. And in this particular group of proteins, bromodomains play a very important role. So my group, and also many other groups, have been interested in one particular protein belonging to this group, to this bromodomain group, that is called CREBBP. And as you can imagine, as synthetic organic chemist, if you want to interfere with this process, the goal is to be able to design small molecule inhibitors that will mimic the natural ligand, in this case, this acetylated lysine residue, and compete with it so that we can block these recognition pathways and therefore have a way of controlling the biological response. So in this context of developing small molecule inhibitors, as I said, in our case, we use computational tools and molecular dynamic simulations in collaboration with my colleague Amadeo Cafriche. We found that a relatively good inhibitor for this type of interaction would be an oxyindole molecule bearing a CF3 group in this position here. So keep in mind, I just go back, that we are trying to mimic this acetylated lysine residue, and this is why our small molecule also contains this N-acetyl group on the nitrogen of the indole, which is going to be kind of the mimic of, or the competitive binder for the natural ligand. Okay, so we were here finding through our computational studies this kind of molecule and asking ourselves how we could then do medicinal chemistry connected to this molecule, how we could make different derivatives of this molecule in a straightforward manner. And this is the beauty of organic chemistry and methodology development, that sometimes discoveries that we might think are not going to be relevant, they become relevant down the road. So in our case, fast analysis of existing literature showed us that trifluoromethylation reactions of N-vinyl substituted amides, N-aryl vinylamides, could be a good platform to access the kind of moieties that we were interested in. And there was, at that time, a couple of interesting reports by the group of Woshen Liu and Mikiko Sodeoka on this type of chemistry. So what we set out to do in my group was to apply existing methodologies onto a potential substrate and then see how flexible the chemistry would be in order to make different derivatives. So to do that, we decided to protect the nitrogen of the precursor with an aryl sulfonyl group. And this is how our starting material looked like. We decided to then expose this starting material to the conditions initially reported by Mikiko Sodeoka, which involved the use of this trifluoromethylating agent, which is Tony reagent, 1.2 equivalent, in the presence of a soluble copper salt, copper 1 salt, and a bipyridine ligand. And applying these conditions that were already reported, as I said, we were happy to see 50% conversion to something that we assumed would be the desired product, so the product in which the CF3 group has been added to the terminal position of the alkene, and the resulting intermediate at this position here, in principle, maybe a radical intermediate, could be intersected by the neighboring aromatic group to form this new carbon-carbon bond and close the corresponding oxyindole. However, after a careful analysis of this reaction, we realized that this was not the product that we had made, and that indeed the product that we had synthesized under these conditions was this amide shown here. And it was a remarkable transformation in our eyes, because if you see here, the connectivity of the molecule has changed quite a bit. So we have removed the SO2 group from the molecule, and we have specifically connected these two carbon atoms that were in remote position from one another in the starting material. We have bonded them, so put them together in the structure of the final product. So if you look at the starting material and the final product, then you will realize that these things have happened. We have trifluoromethylated the double bond, so we have introduced the trifluoromethyl group at this position as we expected, but we have also desulfonylated the substrate. Formally, this aryl group has migrated in a 1,4 manner, and in this migration, a new carbon sp3, carbon sp2 bond has been formed. And then, of course, we ask ourselves the question if this chemistry was general. This is already a while ago, back in 2013, but we realized that indeed the chemistry is general, and just a small modification of the protocol, still using Tony reagent, but here using copper oxide as a catalyst, afforded a wide variety of substituted trifluoromethylated amides, alpha trifluoromethyl substituted amides, in reasonable yields, I would argue. So keep in mind that all the examples here, I only show you 4, but we had many more examples. All the examples contain an aryl group bound to nitrogen, okay, in our starting material, and of course into the final product. So we tried to extend the scope of the chemistry to N-alkyl substituted substrates. And when we did that, we were extremely pleased to find that what we obtained were the corresponding oxyindoles. Again, these oxyindoles do not stem from the direct trifluoromethylation and reaction with the aromatic group, because if you look at the structure of the final product, again, the SO2 molecule, an SO2 molecule has been lost. So, in other words, this oxyindole seemed to be a product in which this type of process has taken place, this formal aryl migration has taken place, but additionally, a carbon-nitrogen bond formation has taken place to form the corresponding bicyclic structure. And again, the chemistry was quite general, and we could also utilize this type of transformation for our medicinal chemistry campaign. Beyond the medchem interest into this type of molecules, we became interested in expanding the scope of these transformations to other type of residues that could be incorporated into the structure of the final compounds. I have showed you how trifluoromethylation reactions work, but using this acido version of Tony reagent, we realized that we could incorporate acido groups into the structure of our amides or oxyindoles, or combining phosphinic acids and silver salts. We could also introduce the corresponding phosphonates into the structure of those final products. And these are just extensions of the same chemistry. But mechanistically, all these transformations we thought could be rationalized following this scheme that I show you here. Under different set of reaction conditions, we identified the formation of key radical intermediates in the reaction that would undergo addition onto the corresponding vinylamide double bond. This radical addition would generate this alpha keto radical intermediate that we generate a radical in situ, addition to the double bond takes place, and formation of an alpha keto radical intermediate occurs. This alpha-keto radical intermediate now interacts with the ipso carbon of the aryl sulfonyl group that is protecting the nitrogen to form a spirocyclic intermediate that you see here. This is non-aromatic, and therefore to recover aromaticity, this will be the driving force to release SO2 and generate an amidyl radical intermediate, whose further reactivity depends on the R group that you see here. So maybe before I go to the reactivity of the nitrogen center radical, let me just remind you that this kind of transformation in which formally we have had an aryl migration reaction, in this case radical mediated, can or resembles the classical Smiles rearrangement reaction, in which, for example, biaryl sulfones can be treated in the presence of a strong base, generating a negatively charged benzylic intermediate, triggering the migration to give this type of products. And if you see this kind of chemistry that is here polar and based on a carbanion intermediate, resembles very much this same reaction that you see here, only that here it takes place via radicals. So as I was pointing out, then we have a nitrogen center radical intermediate, and then the reaction will proceed via hydrogen abstraction to form the corresponding amides, or in the case of N-alkyl substituted nitrogens, there will be a reaction between this nitrogen center radical and the aromatic ring in ortho position to form the additional carbon-nitrogen bond, and then upon aromatization, we would obtain the corresponding oxy-indoles. So you can see that the mechanism is kind of divergent. So from a common intermediate, we can access two different products depending on the substitution pattern on the nitrogen atom. And this is kind of the summary slide where you see all this chemistry put together: radical addition, formation of a spirocyclic intermediate, generation of an amidyl radical, and then formation of the products. We did different variations of this chemistry. As you can imagine, we aim to harvest this nitrogen center radical, for example, by introducing in ortho position of our substrates an alkyne. In this case, you can imagine that the same type of chemistry that I have presented you before, up to the point of the generation of this nitrogen center radical, would bring us to this type of intermediate here. And now the reaction with the neighboring alkyne would deliver a vinyl radical that again can intersect this aromatic ring, forming this four-member ring structure. Another variation of this chemistry is to modify the substrate with a vinyl group in the ortho position. In this case, you have two double bonds that will compete for the radical. The vinyl group, so this styrenic double bond, will be more reactive, and therefore the chemistry will start there. Addition of the radical to this position will generate a benzylic radical that would now undergo an 8-endo-3 cyclization and formation of, again, the alpha-keto radical intermediate, ipsocyclization to form this very encumbered intermediate, rearomatization with loss of SO2, and then formation of the corresponding indene derivative with good levels of relative stereocontrol. So only one diastereoisomer is obtained in this position. So these are just different variations of this chemistry, and we think the community kind of picked on this idea of utilizing radical protocols to trigger the migration of aryl groups onto different positions of the molecules. So these Smile rearrangement reactions, I just show you here a couple of examples, both of them from the group of Corey Stephenson, but many other people, as beautifully summarized in this review by Mike Greeny, have really contributed in this research area. Now, all these transformations, radical mediated, after a while became, as I said, popular, but there were no asymmetric variants. it was not possible to make them asymmetric. This doesn't mean that there are no smile rearrangement that can be done asymmetrically. And in fact, the group of Jonathan Clyden has showed for a number of years now that chiral ureas, again, via polar intermediates upon deprotonation with a strong base, can undergo, can trigger the migration of aryl groups onto the alpha position of one of the nitrogen atoms with complete retention of stereochemistry with absolute stereo control, and then cleavage of the urea can deliver the corresponding chiral amines in excellent EEs. So there are asymmetric versions of the polar Smiles rearrangement reaction, but they require the use of strong bases and cryogenic conditions, and it's very important, or is quite key, to have this urea moiety in the starting material, so that at the end we have here a quaternary center bound to a nitrogen atom. So in our case, we were eager to see how the chemistry that I have showed you before could be rendered asymmetric, also thinking about the applicability of this type of structures towards the synthesis of biologically relevant molecules. Some examples shown here. We have a number of natural products containing oxyindoles. Helsemine is a representative example, but there are many more. We have a small molecule NK1 and NK3 antagonists that are applicable for the treatment of gastric disorders that again bear this type of all carbon quaternary center alpha to an amide, as you can see here. The same for the CCR5 antagonist, which is used as an anti-HIV drug, featuring also chiral amide with an all carbon quaternary center. And making all carbon quaternary centers in asymmetric form is per se in organic chemistry an outstanding challenge. You have a lot of steric congestion around the carbon center, and you have, in the case that you are aiming to do so in an acyclic system, you have a large number of degrees of freedom, so it's quite difficult to impart absolute stereo control for this type of moieties. But we thought that we had a relatively good template to do that. So our idea was whether we could replace the sulfone, the sulfonyl group, protecting the nitrogen, with a chiral connector, so to say, a chiral auxiliary, and hoping that we would be able to again generate radicals in the reaction media, and these radicals would undergo addition onto the double bond, triggering the same type of intermediates and the same type of reactivity. But then if our connector between the nitrogen and the migrating aryl group would be a chiral unit, then we would be able to have to impart the stereo control in the formation of this new carbon-carbon bond, and therefore in the formation of this new quaternary center here. One of the key ideas that we had, of course, was to use light to generate those radicals rather than the previous procedures that I showed you before to generate the CF3 or the acido or the phosphoryl radicals. We thought that the use of light would offer us much milder conditions, and therefore we could access a larger variety of radicals in these transformations. And as chiral auxiliary, after a lot of back and forth, we somehow came to the conclusion that sulfoxides could be a good chiral auxiliary for this purpose. First of all, in these transformations, the sulfoxides would work as a traceless chiral auxiliary, so they will disappear from the structure of the final product. You don't have to add an extra step to get rid of it. As I said, the use of light would be compatible with the chiral sulfoxides and will give us mild reaction conditions. We could access these all carbon quaternary centers, and because with the sulfoxides we can access both R and S enantiomer, we could have also both enantiomers of the product. So that was kind of the design. And then of course, when you have a reaction design, you have to be able to make your starting material first. And it took us a little bit of time to figure out the synthesis, and this is why I want to include it here. Basically, we have to make a resolution of this menthol derivative, and then engage the corresponding chiral, the corresponding salt, with the protonated amine, followed by acylation. This is the procedure that we follow for, let's say, 90% of the substrates. In some cases, this chemistry doesn't work or is not robust enough, and we don't obtain the corresponding compounds with good levels of EE. And this is why we have now devised an alternative methods in which, instead of acylating with the corresponding acid chloride, we do the acylation with this mixed anhydride. But in any case, using these routes, we are able to access our chiral starting materials, and then it became the question which type of radicals we can add and whether or not this hypothesis would work. And one of the easiest precursors that we thought about were aryl sulfonyl chlorides. They have been, it has been shown that aryl sulfonyl chlorides can react with iridium photocatalysts to generate the corresponding sulfonyl radicals, and therefore we envisioned the generation of the sulfonyl radical and the incorporation onto the double bond, triggering the reaction cascade to enable the formation of the corresponding amides, bearing an aryl sulfonyl group with absolute stereo control at this position. And I show you here the scope of the reaction, but more importantly, all the examples that I show you here are different aryl groups on the sulfonyl chloride. The most important thing is that one of these compounds enabled us to confirm the stereochemistry of the final products, which was very nice. And as you can see, the chemistry is quite robust, both in terms of yields and also in terms of the levels of stereo control, of absolute stereo control of the corresponding amides. We always get very good enantiomeric ratios. So not only aryl sulfonyl chlorides, but also heteroaryl sulfonyl chlorides and alkyl sulfonyl chlorides are compatible with the reaction conditions. So I won't spend that much time here. Maybe it's more interesting what we can tolerate in terms of the substituents on the nitrogen atom. So you can see that we can tolerate both aryl substituted and, in this case, alkyl substituted. Here we don't obtain the oxyindols. We always go and we always form the corresponding amides. And in terms of the aromatic groups that can migrate, right? So this is also quite an important parameter. We can tolerate aromatic groups with bearing carbon bromo bonds that can be used for further functionalization, as I will show you in a minute. Naphthyl ones, so even larger ones, can migrate, and also heteroaryl sulfoxides are compatible with our reaction conditions. However, the most important part, or the one where we wanted to have more flexibility, was in the nature of the radicals that we can incorporate in these reactions. And this is why we always aimed at using light as our source, right, for radical generation. And I just show you here that this is indeed the case. So we can introduce all sorts of perfluoroalkyl radicals, as you can see here. So radicals bearing trifluoromethyl groups, but also perfluoroalkyl groups. And then we can go from very electron deficient radicals to more electron rich radicals, as you can see in this trend here. So we can incorporate tertiary carbon centered radicals, even non-functionalized ones, acyl radicals, as you can see here, and even radicals generated in the alpha position to the corresponding of the corresponding lactones. Again, here with moderate diastereoselectivity, but still with very good levels of absolute stereo control. And it's important, I think you might have seen it throughout the slides, to highlight that when we start with the S-configured sulfoxide, then we always get the R-configured product. And I will discuss this a little bit in a minute when we go to the mechanism. But before I discuss this, I just show you that indeed the reaction is stereospecific, so that we start with the R enantiomer of our chiral auxiliary, then we obtain the opposite enantiomer of the final product, as you can see here. And then I also want to highlight here a couple of applications of this chemistry in terms of what can we do with the products that we form. So certainly these amides there in this alpha all carbon quaternary center can be transformed into the corresponding amines upon reduction. Then we can also hydrolyze the amides to the corresponding carboxylic acids, which are also very interesting building blocks. And there are not that many methods to make carboxylic acids with an alpha all carbon quaternary center. And more importantly, we can access the corresponding oxy-indoles via a classical palladium cross-coupling reaction when our migrating aromatic group has a carbon bromo bond in ortho position. This enables the synthesis, an enantiocontrolled synthesis of oxy-indoles, as you can see here. So enough about the scope and the applications of this chemistry, and a few words about the mechanism. So it's clear that in the presence of light, our iridium photocatalyst is excited, and it's this excited state of our photocatalyst that is able to generate the corresponding radicals. I have showcased here the calculations, the DFT calculations for the phenyl sulfonyl chloride precursors. But this is certainly the case for all other radicals that I have showed you so far. So the radicals are generated, and then addition onto the corresponding terminal position of the double bond takes place with a relatively amenable activation barrier. As you can see here, the process is slightly endothermic to produce the alpha-keto radical intermediate. Now, this alpha-keto radical intermediate engages with the ipso carbon of this aromatic group, if you remember, via this spirocyclic intermediate, non-aromatic, to produce the corresponding, in this case, SO center radical. And what is interesting is that in this step, right, in the formation of this spirocycle that then undergoes ring opening to form this intermediate 3 shown here, is where we define the absolute stereochemistry of our new stereocenter of this carbon atom here. And very simple, a very simple model allows us to explain why we go from an S-configured sulfoxide into an R-configured product. So typically, it's much more preferred to have the methyl group or the group in the internal position of the double bond facing the aromatic group that is going to migrate into this intermediate, as compared to having so to say, the terminal position, the substituent in the terminal position of the double bond facing the aromatic group. So I show you here the 2 Newman projections. This is much more favored, and this is the one that determines the absolute stereochemistry. Now, from this intermediate 3, we have to close the photocatalytic cycle, and we do so by oxidizing the SO, but we never detect SO2 or SO in our GCMS. What we detect is always the formation of KHSO3. We always have a base in the media, and we think this is how the sulfur byproduct of the sulfoxide is kind of released into the media. And then, of course, closing the catalytic cycle delivers the desired products. So this, so translating the chemistry of a radical addition onto a double bond into the formation of chiral amides or chiral oxy-indoles is something that took us quite a number of years, I have to say. But indeed, at the same time, it spiked our interest in kind of taking a step back and simplifying things. You have seen that all our starting materials are quite tailored, right? So we need the starting materials that require multiple steps, that are carefully designed to orchestrate the chemistry that we are interested in. And at some point, we started to think whether we would be able to use the knowledge that we had in generating radicals and the addition of those radicals onto alkenes to develop difunctionalization reactions of alkenes in which we could merge both transition metals and radicals in one pot. I think this field today needs no introduction. So the number of groups and the ability of those groups to enable this kind of difunctionalization reactions on alkenes is now out of question. So there are many different ways in which we can do that. I would argue, and I have tried to summarize here, of course, in an oversimplistic way, the type of olefins that have been typically used for this type of transformations. You see olefins that have somehow a kind of chelating group close to the double bond, or olefins that are activated by a conjugated aromatic ring, or activated by an electron withdrawing group, or even olefins that contain a kind of pseudo chelating group, for example, an acid, so that you can form a transient directing group and then get rid of it. But what is common in all these processes, regardless of the type of olefin that they use or the metal that is used to orchestrate these reactions, is that typically both a nucleophile and an electrophile are added across the double bond, taking advantage of the natural reactivity of olefins as dipoles, right? And what our group kind of decided to do was to take a step back and then think about whether we would be able to access different olefins and also different reactivities if we would engage two different electrophiles adding across the double bond. It is very well known by the group of Greg Fu and many others that electrophilic couplings catalyzed by nickel can form highly challenging carbon-carbon bonds between two electrophiles, of course. And our question was whether or not these two electrophiles, instead of undergoing the direct coupling, could be intercepted with an olefin so that formally we could difunctionalize the corresponding double bond. In this case, we were specifically interested in dicarbofunctionalization reactions, adding two new carbon-carbon bonds across the pi system. And of course, we wanted to do so with all sorts of olefins, both olefins having directing groups and olefins having no directing group whatsoever. And I show you here how we managed to identify the right reaction conditions for this reactive dicarbofunctionalization reaction of olefins using a nickel-2 pre-catalyst, this nickel dichloro tetrapyridino complex, in the presence of a diterbutyl bipyridine ligand and TDAE, which is this organic reductant shown here. We managed to selectively add two different electrophiles, a carbon sp2 electrophile and a carbon sp3 electrophile, across the double bond of olefins to form these dicarbofunctionalized products. Both in a chemoselective fashion, keep in mind that these two electrophiles could couple together, but we do not observe the product of this direct coupling. Then also in a regioselective manner, since here we have two different electrophiles, then the question is which electrophile is going to be added in the terminal position and which one in the internal position. But we only get this type of products in which the sp3-based electrophile is added at the terminal position and the sp2-based electrophile is added in the internal position. And of course, the question mark is about the stereoselectivity, and this I will discuss now in a couple of slides. But let me first show you a little bit the scope in terms of olefins that can be tolerated in this chemistry. You can see here that we can tolerate olefins having electron withdrawing groups, so partially activated olefins, as the one that I shared with you before, and other groups have also shown that they are compatible with dicarbofunctionalization reactions. Interestingly, here is an example of a 1,2 disubstituted olefins, which are typically not very well behaved in these difunctionalization reactions, but for us, they were quite well behaved with very good levels of diastereo control. We can have allylic systems, allylic alcohols, allylic amide, or allylic phosphonates as well. And you can see here also a little bit of scope in terms of aromatic groups that can be incorporated. We can incorporate aromatic groups bearing carbon boro bonds or carbon bromo bonds that do not undergo subsequent reactions, but one could think about functionalizing them farther, carbon chlorobonds. And then in terms of the sp3 electrophiles, you can see here, I show you a few examples, but all of them are tertiary. That's a very important thing, a very important feature of this chemistry, which you can see it as a limitation, but on the other hand, it's quite difficult to actually install carbon-carbon bonds in such congested systems. So for us, this was rather an advantage more than a disadvantage. But as you can see is that in all our olefins, we needed a kind of coordinating group, or at least a group containing an heteroatom close to the double bond for the chemistry to work. And it took us, so this was 2017, and it took us almost a couple of years to figure out how to adapt the protocol so that we could incorporate now olefins containing no directing group, no coordinating group, no activation whatsoever. So truly unactivated olefins. And the trick was to change the nickel precursor, keeping both the ligand and the reductant, and changing the nickel precursor and changing also the solvent in the reaction. So a little bit of the mechanism of these reactions. I showed you, or I explained you, that one of the competing reactions could be the coupling between the two electrophiles together. And of course, another potential side reaction could have been that our alkylic iodide would undergo, in the presence of nickel and a reductant, an ATRA process in which we just halogenate, and so we do the incorporation of the halogen and the carbon across the double bond, and then this intermediate iodide undergoes cross-coupling with the electrophilic sp2-based partner. In other words, we could explain the formation of the observed products via these two steps, but all our control experiments seem to rule out this idea. And in fact, what we see is that when we prepare these potential intermediates, these secondary alkyl iodides, and we subject them to our standard reaction conditions, they never deliver the corresponding cross-coupling products, so that we rule out the formation of alkyl iodides as intermediates in this transformation. Then, looking at the nickel species involved, we did some control experiments that nicely showed how under, first of all, that nickel zero species could undergo oxidative addition onto the corresponding carbon sp2 iodide bond to form aryl nickel 2 complexes. This is well-established chemistry. What is even more interesting is that if you have a nickel 2 precursor and you expose it to TDAE as a reductant, then you get the same type of compound, formally showing you that TDAE, our reductant, seems to be able to bring nickel 2 species to nickel 0, so that then oxidative addition towards the formation of nickel 2 is possible. And having these aryl nickel 2 complexes in hand, we use them stoichiometrically under our reaction conditions in the presence of olefin and in the presence of the alkyl electrophile. And we were happy to see that they could indeed transfer the aromatic group into the structure of the final product with very good levels, with very good yields. So these aryl nickel 2 iodide complexes are competent intermediates in our reactions. So even if this is an oversimplification, what I'm going to show you here, we rationalize this chemistry thinking we have a nickel 2 precursor that in the presence of a strong reductant such as TDAE is going to be reduced to nickel 0. Oxidative addition onto carbon sp2 iodide bond is going to be rather favored to form this aryl nickel 2 complex. And again, we have experimental evidence for the fact that this aryl nickel 2 iodide complex is a viable intermediate in the reaction. Somehow our carbon sp3 electrophile should be the source of an alkyl radical that undergoes addition onto the terminal position of the double bond, and then these two intermediates come together to form this key aryl alkyl nickel 3 iodo complex that undergoes reductive elimination to form the corresponding bicarbofunctionalized products, right? And we have seen that these aryl nickel 2 precursors are competent, as I showed you before, And then to close the catalytic cycle, we need to then propose that are these nickel-1 species formed in the media, the ones that are responsible for the activation of our sp3 electrophile. Many more computational work, mechanistic work, kinetic work on this system has been done, and the mechanism is by now refined, but at least here I just leave you with some key mechanistic aspects of this chemistry. The most important one maybe being the formation of this key intermediate as the one preceding the formation of the product and generating the catalytically active nickel-1 species so that the catalytic cycle is closed. And for me, it's important to highlight this intermediate because somehow this intermediate is the one that is going to give us an entry into the potential development of an asymmetric version of these reactions, right? So if we believe that the product stems from this intermediate, then utilizing chiral ligands on nickel should in principle allow us to have absolute stereo control in the formation of the final carbon-carbon bond in these reactions. As you can easily guess, the next thing that we did was to take some of the substrates that were working well in the previous transformations, our two electrophiles, the butyl iodide and iodo-benzene, with our nickel-2 precursor and our organic reductant, TDAE, and then explore chiral ligands that are well known to impart absolute stereo control in nickel-catalyzed reactions. I have to say that again here, which I think for those of you familiar with nickel chemistry, it will come to no surprise, we found very difficult to rationalize results. So we had ligands that could be quite lousy in terms of efficiency, or others that would be very well behaved in terms of the yield, in terms of conversion, but then the EEs would be very poor. And finally, these bisoxazolene ligands, as you can see here with an isopropyl group, kind of showed us the trend in the right direction. So the presence of an isopropyl group enabled us to isolate the product of the dicarbofunctionalization reaction in 64% yield and 88% EE. And with a little bit of further optimization of the side chains in the bisoxazolene ligand, then we could come up with a ligand that would give us both very good yield and very good EE. And I just show you here the scope of this reaction. I will not invest that much time because the chemistry is relatively simple. So we just dicarbofunctionalize vinylamides with aromatic iodides and bromides also in the presence of tertiary alkyl iodides. You can see here the scope, both in terms of the aryl iodide or aryl bromide. We can tolerate heterocycles. We can also introduce vinyl groups, which is quite interesting for further manipulation. In terms of the alkyl iodides, this is always our main theme. Only tertiary ones work well in this chemistry. This is why the diversity there is relatively limited. In terms of the vinyl amide acceptors, we can tolerate different kind of amides, aromatic ones, and also alkylic ones. And what is maybe more interesting is the type of olefins, because not only vinylamides are compatible with this protocol, but also vinyl boranes that can be further functionalized into the corresponding secondary alcohols, adding an oxidation workup, an oxidative workup, and also vinyl phosphates could be nicely difunctionalized in asymmetric fashion with this chemistry. And just one last spin-off from this chemistry is to replace one of our electrophiles with a silane as a reductant, so that instead of generating an aryl alkyl nickel species, we would generate a nickel hydride that would then do the insertion onto the double bond, formally resulting in the corresponding asymmetric hydroarylation reaction. So this was interesting for us in terms of the type of molecules that we could access. And this is why, as you can see here, with a slight modification of the protocol, and a slight change also again in the ligand. In this case, a bis imidazole ligand was needed. We could asymmetrically hydroarylate a large variety of amides, of vinyl amides. I show you here the scope in terms of the aryl groups that can be incorporated, and of course the further functionalization using classical palladium cross-coupling reactions. And I show you here the scope in terms of the vinyl amides that we can incorporate, in some cases with moderate or low yields if the olefin site are difunctionalized. Most of them are terminal, but if we difunctionalize them, then efficiencies suffer, but you can see the levels of absolute stereo control are still very good. And in terms of mechanism, as I said, the chemistry is quite related. So one has to, again, envision a nickel-1 iodide as the key intermediate that undergoes ligand displacement with the silane to form the nickel hydride, which inserts into the olefin with this particular regioselectivity. We, of course, computed the opposite regioselectivity for the insertion, but the energy is relatively high for this process. And then this nickel-1 intermediate undergoes oxidative addition with our aryl iodide or aryl bromide to form this aryl nickel alkyl aryl nickel 3 complex, which undergoes reductive elimination to form the corresponding product. And imparting absolute stereo control again comes from this intermediate here where reductive elimination takes place, and we also could characterize the transition state that precedes this reductive elimination. So that interestingly we can see that indeed for the reductive elimination to take place, one of the nitrogens of our chiral ligand is very much elongated from the nickel, so that the nickel can host, so to say, enough space for the reductive elimination to take place. So I don't want to take more time on the chemistry. I want to close, hopefully having been able to show you how the interaction of radical metals and alkenes can give us a lot of interesting chemistries and transformations that we have in the last few years aimed to do in asymmetric fashion. And of course, the last couple of slides is for me to thank the current group members. I'm extremely blessed to have this group of individuals working with me, putting all their effort and all their minds and passion into the chemistry. I have to say it's a very lively group of individuals, and I'm extremely grateful that despite the difficult circumstances in the past months, they have been able to keep the stamina and move forward. But certainly I would not be here today, of course, without them, but without all the previous ones that have been part of the Nevado family or the Nevado group over the past 12, 13 years. We always had a chance to do fun activities together, which I hope we will be able to do again in the near future. I have to say, people say it does take a village to raise a child, and it does take a village to raise a group. So I'm extremely grateful to all of them, so past and present members. And of course, I cannot close this talk without thanking Kime and the Science of Synthesis for thinking of me for this award. Margaret, because ever since I met her in the board of editors of Organic Synthesis, I realized what a role model should be. I have to say, she's one of the most inspiring chemists I have met throughout these years. And being able to get this award from her, and also being the second recipient after Sarah, who I considered a very good friend and also a chemist I admire a lot, is really a true honor for me. And finally, I would like to dedicate this talk and this award to my late father, who passed away just very recently. And even if he didn't understand a single word of chemistry, I can assure you he would have been extremely happy and proud of me today. So thank you very much, all of you, for your kind attention. I don't know if there will be time for questions. I typically go over time. I apologize for that, but thank you very much anyway.